We at Sinai like to save the best for last. Thank you for staying. It's time for some nanomedicine. We have two fantastic talks upcoming. First up is Dr. Michael McMahon from Johns Hopkins School of Medicine. His background is physics and physical chemistry. He specialized in NMR spectroscopy. Then turned to biomedical imaging at a postdoctoral at MIT before moving to Johns Hopkins finally, where he's an associate professor. And today he'll be talking about some organic MRI contrast agents, which is I'm particularly excited, away from iron oxides and gadoliniums. So without further ado, floor is yours.
Thank you, Carlos. I'd like to thank the organizers for giving this opportunity to speak about my work. I work on developing alternatives to gadolinium, which are purely organic. So first, just like to mention what an MRI contrast agent, what is it good for? Well, we hope to develop contrast agents which can be introduced into a functional region or anatomical region to alter the MR signal. And of course, most widely used are the T1 contrast agents based on gadolinium. And so what you see from these images at left, here's a T2 weighted image, a T1 weighted image. A little bit difficult to find the tumor. However, when you administer gadolinium, it pops quite nicely out. And it's also quite frequently used for MS lesions and other types of tumors. One of the challenges with gadolinium is that it's a toxic in its free form, but it's approved to administer to patients when it's chelated by a couple different types of groups. And so there's been a large effort because at this point 30% of all MRI scans require administration of gadolinium chelates, and so there's both macrocyclic and linear chelates. However, a few years back, Henrik Thompson and some others noticed that patients would get this nephrogenic systemic fibrosis in the kidneys associated with gadolinium administration. And so, particularly for patients with chronic kidney disease or some other sort of challenges, it's not ideal to administer gadolinium. And in addition, others have shown that gadolinium chelates will accumulate in brain tissue over time, which would raise the risk of gadolinium release and then more challenges. So based on this, we felt that alternatives should be developed. So what we're doing is we're working on chemical exchange saturation transfer, and this is a different type of contrast mechanism, and it allows not the use of gadolinium. So for standard MRI, you might have a spin echo or a gradient echo acquisition scheme. For CEST, we need to place some sort of preparation period to prepare the magnetization. And in this case, what we do is, for CEST, we destroy the magnetization of these solute protons. And if there's a rapid chemical exchange of these solute protons with bulk water, you'll then see first the solute labile proton magnetization drop, and then this transfers the signal loss to water. And so if this exchange is rapid enough and still also tuned appropriately, the chemical shift being appropriate, then you can see very large amplifications of this signal onto the water, which is a nice feature for contrast agents. So this is a growing field. It was introduced in 2000 by Robert Balaban at NIH. By 2010, we had our first international CEST workshop with 49 attendees. I held one in 2012 with 82, and now it's grown to over 200 attendees as of last year. So that's not 2012. And the citations have been growing in this area as well. So one of the first things that people notice is that cancer cells, when they're perfused, you detect a large number of rapidly exchanging amide protons. And so what they thought of is, well, we can do a high-res NMR, and we have to play all these tricks to detect these protons. Maybe we can actually go the reverse way and destroy the magnetization of these labile protons and then look at it on water in tumors. And so in our center, they first prepared some 9L tumors in rats, and when you place the saturation pulse and sweep it across the chemical shift spectrum, you see a nice peak at around 3.5 ppm, 3 to 3.5. And so when you collect this CEST image at that position, you can see the tumor highlight very nicely, and the highlighting compares well with histology. Because this is just involving a simple additional saturation pulse on a standard MRI scheme, you can use the same hardware. So within 3 years, from 2003, they presented their tumor results in rodents. By 2006, we actually had it implemented on our clinical 3T scanner. And so now we can actually collect CEST images on patients with brain tumors and see very nice highlighting regions. So again, those were more MR physicists, and I had an interest in developing good compounds which actually could be used to using CEST imaging to detect. And so we started out with some simple organic compounds, some sugars, some polyamino acids. These were able to detect relatively well on high field preclinical scanners. But we really were interested in developing compounds which we could detected at 3T. And so because our site about 10 years ago had the first patient images, fortunately Philips Healthcare spent some time and upgraded our systems so that we could have these very long saturation pulses and then alternate between the two different amplifiers in a parallel transmit channel so that we can destroy the magnetization over several seconds to get very nice CEST imaging. So this is now a system that's on about 6 sites in the world, and Philips Healthcare helped us develop this. And this is what we would like to use. We would like to optimize our compounds for detection on these scanners. So what are the optimal properties to detect this at 3T? Well, I performed some spin block simulations to characterize this first. And so what you see is if the chemical shift of the labile proton approaches 10 ppm from water, and if the exchange rate is on the order of 500 hertz to 1,000 times per second, then you can see very nice enhancements compared to the hydroxyls, about 1 ppm, or the amide protons, about 3 ppm. So those were the kind of characteristics we were looking for. Another interesting study is a group in Torino showed that the NH protons on iopamidol here resonate a little further from water, about 5.5 ppm. And so this is a nice compound because actually this is something that is used as a CT contrast agent right now. So it's been approved for high dose administration into patients, and clinical experience in administering this compound since 1986, so a long time. And so they have been working on developing this as a CEST imaging agent. And so based on their work, we knew that we should probably have some aromatics and heterocycles, which might in our compounds. And so now we finally, after a few years of looking around, have come up with some designs that work well. And so here's our first design that's really very nice, and it's based on salicylic acid. Now you guys might know salicylic acid because it's basically been used for a long time in pain treatment. So the acetylated version is aspirin, and so for about 2,400 years this compound's been in man. And so how would it work as a CEST agent? Well, it has one labile proton, the OH. And unfortunately, if it's just a phenol proton, it exchanges way too fast with water to actually destroy the magnetization. However, when you place this carboxyl right next door, what happens is you induce a shyness in these protons. And so they exchange much, much slower with the neighboring water based on this low barrier hydrogen bond. And this also increases the chemical shift of these protons. Another nice feature of these compounds is that you have this ring. There's 4 additional tunable sites. So you can use all these different sites to try and fine-tune the shift and also the exchange rate. And so this allowed us to do a nice systematic study of what factors might improve CEST contrast. So the first thing we looked at is playing around with the hydrogen bonding site. So is the carboxyl the best site? So unfortunately, when the hydrogen bonding is too strong, you see almost no CEST contrast. And unfortunately, we haven't found another hydrogen bonding partner that was in the right tight range like the carboxyls. It's either too strong or too weak. We then next looked at modifications to these R4 and R5 sites. And so what we notice is that when we place hydroxyls, carboxyls, sulfuric acid groups nearby, the shift and exchange rate are relatively unmodified. So these are very nice sites in terms of if we want to conjugate here, it's not going to produce a big problem for the contrast here. You can use a very large acidic group and change the chemical shift a little bit and also increase the exchange rate. However, if you start to modify the R6 position, what you see is very, very large changes in the hydrogen bonding and hence the chemical shift or chemical exchange rate transfer. Same thing with the R3 site. This is a site that's very, very sensitive, so we have not changed those, but they could be used to turn the contrast on or off. So based on our study over a series of papers, we found these CEST agents which had chemical shifts and exchange rates that fall in this nice sweet spot. And a nice feature of these is, again, a lot of these are members of non-steroidal anti-inflammatory compounds. So these are good compounds that are used by pharmaceuticals now. And also, some of these are natural metabolites of cherries, cranberries, and wine grapes. So they're fairly safe compounds. We did perform some cell culture assays of toxicity for 19 of our favorite agents. And we saw that they were relatively well handled by HEK cells, even at millimolar concentrations. And so here's just a couple that work very, very nicely and are also extremely safe. So now that we came up with a really nice MRI contrast agent, what I'd like to do is develop theranostics based around this salicylic acid. So the idea is that MRI agents can be used to highlight where therapeutic agents are delivered. And a nice feature of these agents is that because the chemical shift might differ between different contrast agents, we could actually administer multiple therapeutics and then image where each of these go in one single image. And so we particularly were interested in developing these salicylic acid nanocarriers for imaging of brain tumor delivery. So in particular, what we were interested in first is looking at convection enhanced delivery of nanoparticles to brain tumors. So what is this? In convection enhanced delivery, what you do is you insert a cannula and then you apply a very constant pressure and slowly infuse the compound. And by doing so, you can actually get a very large volume of diffusion for these drugs. And so this is and also minimize the tissue damage. And so this is a nice way to locally administer therapeutics to brain tumors. And of course, there's a large number of controllable variables, and because tumors are heterogeneous, it might be that you have to change your infusion protocol for each patient. And so this is a nice type of therapy to have imaging as a guidance tool. So CED has been used in clinical trials, and it's shown some promise. One compound that I'd like to highlight is this topotecan. It's a camptothecin derivative, and so it's been moved to phase 2, phase 3 trials. However, there's been mixed results into how much benefit the patients have received. And so imaging of the biodistribution of therapeutics for CED should be well justified. And we're not the first group to think about this. Christoph Bankowitz has spent at UCSF has spent quite a bit of time developing these specialized cannulas, which are MRI compatible. Here's an image where you can see the cannula in place and the infusion area, which can be monitored in real time. Again, gadolinium infusion could be used but what if multiple positions of the cannulas are required? These CEST agents should probably have some additional benefits in this particular case. So our first type of nanocarrier, and this is the one we're focusing on right now, is we use dendrimers. So these are these dendritic polymers with a large number of surface conjugatable groups, and they have been administered and shown promise for neutron capture therapy with CED. Very versatile. You can conjugate any number of different types of agents to the surface, such as our salicylates, camptothecin derivatives. And so we'll first evaluate these in a mouse model of GBM. So it's a relatively simple synthesis. We took an amino salicylic acid derivative, had this nice conjugation chemistry, and you can easily get up to 40 groups out of the 126 sites on a G5 dendrimer. For our first designs, we kept the remainder of the surface sites as amines, and what we noticed is that the exchange was much faster than our small molecule salicylic acids, now about 4,500 times per second instead of about 500. So we tried a different approach. We basically attached the glycidol groups to the remainder of the surface, and then we got a nice sharper exchange rate, nice tighter frequency pattern. And if we acetylate the surface, then we can get even slower exchange rates close to the small molecule compounds. And so what's happening, we notice, is that basically these the other exchangeable groups catalyze the exchange of this proton to water, so it basically acts as a exchange catalyst. So next, now that we had our first diagnostic dendrimers, we wanted to try and test it, see if we could detect these in mice with brain tumors. So in this case, we can basically inoculate mice with human U87 glioblastoma cells. We can use the same needle hole to administer the agents as we use to inoculate these tumors. And then in our first case, we just did a very rapid 10-minute infusion of our particles and collect CEST images over time to see how well we can detect these compounds. So here's our first set of images. Here's pre-injection, after injection. Up to an hour and a half later, we still see nice, robust contrast. And the coverage of these particles is about 50% of the tumor, so they don't go across the entire tumor with this infusion rate. But they're well detected and very stable. So now that we had the imaging established, we wanted to prepare theranostic dendrimers. So again, since topotecan was very nice in terms of performance for convection enhanced delivery, we looked at this non-soluble derivative of camptothecin, SN38, which is related to topotecan, which is more soluble. So we attached this to these G5 SA dendrimers, and then tried to see how well these perform therapeutically. So here's just some release and cell toxicity data for these theranostics. You can see that our assay is relatively stable on the G5 dendrimers. However, within 120 hours, basically half of the SN38 is off, which is a nice feature for a therapeutic. And then when we incubate the U87 tumor cells with our G5 SN38 dendrimers, we see very nice tumor killing capability based on this IC50 measurement. So you can see, and in fact, there's another nanotherapeutic NK102, which is a micellar version of the SN38 delivery, and we had about a factor of 3 more potency than that nanoparticle. So we have a nice design. And so we're very optimistic about the use of this nanoparticle. So I'd like to move to a very different area of contrast agent work, and this doesn't involve nanoparticles, but it's of interest probably. So one of the features that CEST is that it's very excellent at measuring pH. And so because it's chemical exchange and the amount of OH or hydroxyl or protons will change the exchange rate, we can use the amplitude of these contrast peaks as a way to detect pH. And of course, tissue pH can be altered in many pathological states, including renal failure and also in cancers. And this has been reviewed by others. There's no really great way for measuring pH in vivo. People have worked for a long time using MR spectroscopy, using phosphorus, fluorine, or proton. However, they're very low sensitivity. And so we thought this is a nice way we can get an amplified detection of our solute and get a better, higher resolution contrast maps than would be attainable with these spectroscopy types of methods. And so for spectroscopy, Sebastian Zerdan spent quite a bit of time, and what he's developed is this imidazole, where this proton here on the imidazole ring is very sensitive to pH, and so they've played around and administered this into a variety of different types of tumors. And so you can see this peak move for different tumor types, and that's how they've performed pH imaging agents. Again, there's no exchange, so there's no amplification of this signal, and so it's very difficult to get a high-resolution map. But they showed that imidazoles have a nice property for pH imaging. And so what we've discovered is that we thought that pH imaging would be particularly interesting for kidneys because they filter waste from the blood and regulate the acid-base balance in the body. And there's not always a great diagnosis for kidney failure using blood tests. And so you could look at alterations in perfusion after administering gadolinium, but as I discussed earlier, it's really not great to administer gadolinium to patients with problems with their kidneys in the first place. So it would be really nice to have some sort of way to measure perfusion and other properties with an MRI agent that's not based on gadolinium. And so, again, the group in Torino has looked at iopamidol, which is the compound I showed earlier, for pH imaging of kidneys. And so here is a group of, and so what they did here is in one group of animals, they administered glycerol to damage the kidneys, and it's kind of a reversible damage. And so at day 3, the damage peaks, and by day 21, it's mostly gone. And so what they showed is when they administered iopamidol in the healthy mice, they saw a very nice, consistent perfusion of the agents. However, for the glycerol-treated agents, you can see very substantial drops in the perfusion of the iopamidol, particularly at day 3, which then grows again. In addition to that, they saw changes in the pH of the kidney, where the remaining pixels that were perfused had a pH of about 7.2 compared to the healthy animals, which with a pH about 6.7. And so iopamidol really showed very nice ways to monitor this kidney damage. However, it's not an ideal agent for clinical 3T scanners. They've also administered this, and I don't show the data, in a occlusion model showing similar nice robust detection of damage of the kidneys. So what we thought is, can we tune the exchangeable NH protons and imidazoles using this hydrogen bonding concept to develop CEST agents? And so our first imidazoles were really not working very well because they had very fast exchange. But then when we built a lot of hydrogen bonding into the system, particularly for this 4,5 dicarboxyamide, then we saw very nice, robust contrast, about 7.8 ppm from water. So this design is something that actually worked out well after a number of different attempts. So here's just our nice pH-sensitive CEST curve. You can see there's two different protons. There's one here and one here, both CEST active. This proton resonates at about 7.5 ppm from water. You can see that the contrast changes very robustly with pH from 6 to 7.5, whereas this proton here, which resonates about 3 ppm from water, is relatively stable. And so this is an agent that should be usable as a pH imaging agent. We did some we did some quantum chemical calculations and showed that, in fact, the reason that this should work as a nice CEST agent is because of this additional hydrogen bonding. That's why the other imidazoles do not seem to work as well. And this is also corroborated partially by some X-ray structures of others. We played around with a few different other options. We added a lysine here. Exchange is a little bit too fast. Added an aspartate instead of a glutamate here. Very similar contrast. So then, just to show you how we actually measure the pH, what we do is we collect contrast both here, and then we take the ratio of the contrast at these two different positions. We do this because then this ratio is concentration insensitive, so it won't be changed by whether or not you have 5 millimolar versus 2 millimolar in a given voxel. And so when you look at this ratiometric map, you can see very nice from 6 to 50 millimolar that we can well detect the pH of these tubes. So we evaluated all three of our agents that we developed. The glutamate, we injected them into the tail vein of healthy mice, looked at the kidneys. The glutamate version showed a little bit better accumulation in the kidneys. And so basically we wanted to try and see if we could create pH maps in healthy mice using this compound. And so we injected them. The contrast peaked about 45 minutes after administration, both at the 7.5 ppm and in the 4.5 ppm positions. And so based on that, we get this nice first pH map where you can see the calyx and then the outer rim, a little bit different pH than the interior. And so the average pH of the kidney was about 6.5, which is very similar to the 6.7 reported by Amy and colleagues with their iopamidol agent. So we are very happy with this compound. So just in summary, we've designed and synthesized a number of different CEST MRI agents which have excellent detection characteristics for clinical 3T scanners. We've looked at both dendrimers of these compounds as theranostic agent and shown that they're detectable with stable contrast in mice. Also looked at them as pH imaging probes, and we have these imidazoles which show very nice pH sensitive contrast. And I'd like to hope that I convinced you guys that this CEST MRI is a new tool for molecular imaging, and it's very exciting. So I'd like to thank all my colleagues point out that we now have our first textbook dedicated to CEST imaging, which just came out in the past 2 months, available on Amazon and through CRC Press. So thank you very much.
I think we have time for some questions from the audience.
Yeah, thank you very much. Really enjoyed the talk. I have just a question. What technique was used to quantify the exchange rate, and in what environment did you perform these experiments?
So what we did is we just measured the pH in PBS, in phosphate buffer saline, and we just used so I published a technique to measure this back in 2008 called where we vary the saturation field strength, and then we look at the
omega plot, basically.
Request, and so that's what we did.
Okay.
But it looks like you can also use this ratiometric method to characterize the exchange rate in vivo.
Thank you.
Hi, great talk. Just have a question on the pH-based measurement.
Mm-hmm.
Since you use such a large ppm shift for your molecule,
Right.
Is it influenced by macromolecule concentration for each subject?
So I don't believe so, but to be honest, what we saw is that we got a similar pH in the mice so far. I'm not sure if there's a tumor nearby, if there might be some additional proteases that might change it, and I don't have any evidence that that's the case at this time.
Thank you.
I actually have a question. Well, whenever we talk about contrast agents for MRI, we all have the question about the dose, right?
Mm-hmm.
How much do you think, at what level concentration do you start to see the contrast?
So CEST agents typically work with about on the order of millimolar in exchangeable protons. Mm-hmm. That's not the same as millimolar in compound, because for hydroxyl-containing compounds you can have 5 or 6 or many more, or for polymers many more copies of the hydroxyl or of the exchangeable protons. So, but it's generally a lower sensitivity technique. It's a slower exchange than the water on gadolinium. Mm-hmm. By definition, you have to slow the exchange rate significantly down from that of the T1 agents to detect it.
Good. So that was actually a very good presentation. I really liked it. It brought me back to my organic chemistry years. So, any more questions? Before you go, we have a small token of appreciation.
Oh.
That's for you. Great. Thank you. Thank you very much.